So hello, first thank you very much for this opportunity to present our research activities and efforts. My name is Špela Miroševič and I'm the co-founder and the president of the CTNNB1 Foundation. So today I would like to present how and why this foundation was created, for which disease, and what are the foundation's key milestones and achievements, and our current research activities. So CTNNB1 Foundation was founded after my son was diagnosed with debilitating disease. My son Urban was born as a perfectly healthy baby boy with 10 fingers on his arms and 10 fingers on his feet. He was perfect in any way, especially when he smiled and looked at us like he knew he was here for a reason. However, not all genetic conditions run in the family. Urban was born with a small typo in his genetic material that occurred de novo. And because of this small typo, he was born with a syndrome so rare that it didn't have a name, but it was named after the gene itself. So CTNNB1 syndrome. And even though the diagnosis crushed us like it would any parent, we were happy to have the diagnosis when Urban was only 9 months old, and the time he was the youngest CTNNB1 patient known. So, to introduce CTNNB1 syndrome, it's a severe and rare neurodevelopmental disorder with first cases described in 2012. Even though there are currently known 400 kids worldwide, probably there are many more undiagnosed. Two recent articles report that mutations in the CTNNB1 gene were found as the most common cause of misdiagnosed cerebral palsy. CTNNB1 syndrome is an autosomal dominant condition caused by mutations in the CTNNB1 gene that occur mostly de novo. CTNNB1 gene produces protein beta-catenin, which has an important role in cell adhesion, communication, and signaling in Wnt pathway. Although the exact prevalence is unknown, current estimates suggest that it affects 1 to 50,000 individuals. CTNNB1 syndrome is associated with intellectual disability and developmental delay, language and speech disorders. Based on our review, there are around 70% of CTNNB1 patients who are nonverbal or speak only a few words. Patients exhibit autism, sleep disorders, have behavioral challenges such as temper tantrums. More than 90% of CTNNB1 patients have facial dysmorphism and visual abnormalities, and in some cases it can also lead to blindness. CTNNB1 kids also have progressive spasticity and axial hypotonia that is present throughout their lives. And of course, the list of symptoms goes on. We are very proud that we recently published a scientific article that may help in the future to look which mutations may be more appropriate for treating with gene therapy and which may benefit with less invasive treatments such as small molecules or maybe antisense oligonucleotides. So this is the first paper published from our team. So when my child received a diagnosis, in the ideal world, we would go to a doctor, the doctor would get us a diagnosis and we would get a treatment. However, unfortunately, we do not live in that ideal world, at least not yet. However, we do live in a time when there is hope with gene therapy. As a parent who is in this situation, hope is something that I desperately need. Hope is something that drives me through the days when I cannot sleep because I worry about my child's future. It gets me through the days when my child struggles to express what he wants. Urban recently turned 3 years. He still can't talk or walk. And although he's perfectly happy this way, we know that speech and motor skills are necessary for his independence. Currently, there are no treatment options, but we are changing this. We have all seen miracles that happened after SMA children were given Zolgensma, or kids and adults with gene-specific eye abnormalities were given Luxturna, or a newer AAV9 therapy called Upstaza for neurodevelopmental disorder called juvenile Parkinsonism changed patients' life. Children who could not even hold their head learned to walk and talk for the first time. So we felt it was necessary to do something to help our CTNNB1 community. There are currently 400 known cases globally with CTNNB1 syndrome and no available treatment. We cannot rely on pharmaceutical companies to initiate the development of such therapies because the number of patients is too small to justify the costs and risk associated with the development. So CTNNB1 Foundation was formed in 2021 to improve the lives of patients affected with CTNNB1 syndrome. Around the same time, a multidisciplinary team of experts was formed and we started our fundraising campaign. We also started the genotype-phenotype study, which we'll present a little later. We started fundraising, and by the end of 2021, we raised first $500,000. We started the development of cell and animal model, and in the mid of 2022, we raised $1 million of funding. We also started our in vitro studies, so testing vectors in neurons and organoids. And currently we have ongoing natural history study and also planned in vivo studies and at the beginning of 2023. In March 2023, we have planned our first international CTNNB1 syndrome conference in Spain. We are also working with the Columbus Foundation and manufacturing company Viralgin, who will assist us in manufacturing process and clinical testing, which is scheduled for 2024. Here we present our research network, which is scattered all over the world. Most of the researchers listed on this slide are presented heavily in our foundation and are very much involved in research activities. Our foundation is funded mainly by the public donations and donations made by the board of directors. So our family donated 16% of the donations that came to the foundation. And additionally, Dr. Damjano Sredkar and Dr. Roman Jerala recently received a 300,000 euro worth grant to support the genotype-phenotype correlation study and investigation of novel RNA approaches. The majority of our donations go to our gene therapy program, which can be seen in the figure, and this is the program that is the further along. Specifically, the development of AAV therapy and that happened at the Children's Medical Research Institute in Australia, and the natural history study of mouse model and the efficacy and the tox study at the Jackson Laboratory. So now I would just like to give you a brief overview of our research activities. So we started our project with the genotype-phenotype correlation study. This study started 2 years ago. It was also published in the clinical trials. We enrolled around 125 patients from 24 countries, and we did medical clinical interviews with them, of course, with our doctors. And we have really robust and good data from this study. And also, Professor Dr. Edwin Chen is currently studying the genetic variants of the patients enrolled in the study with the goal of determining which mutations resulted in a loss of function, dominant negative effect, or maybe protein has some function which leads to a milder symptoms. This study also has a potential to be a natural history study for our gene. And we also are exploring the alternative therapeutical strategies for CTNNB1 syndrome of gene therapy. This is the project from Professor Dr. Roman Jerala and team at the National Institute of Chemistry in Slovenia. So researchers are using a variation of CRISPR-Cas9 techniques, specifically gene editing, to correct mutations at the genomic level. And in addition, a variety of different therapies to induce the upregulation of beta-catenin and downregulation of beta-catenin inhibitors, including components of the destruction complex and various inhibitory regulatory RNA molecules. This will be made by antisense oligonucleotides at the transcriptional level. And the advantages of this approach is adjustable dosing of the drug. And of course, ASO and SRNI have already been used and established in therapy. We have preliminary results which are very encouraging. And of course, our gene replacement therapy, which is the furthest apart. Of course, the invented name is named after my son. It will be called Urbagen. Researchers are using a single-stranded recombinant serotype 9 adeno-associated viral vector. We are currently testing this vector with 2 different promoters. And after our efficacy study, we will determine which promoter shows the best efficacy and, of course, the most safety profile. So the goal of this treatment is to introduce a functional copy of the CTNNB1 gene into the cell, resulting in long-term production of beta-catenin in sufficient quantity to promote neuronal activity and physiological function. So this project has developed more than a dozen therapeutical CTNNB1 gene therapy variants, and some of them have undergone early in silico evaluation and are being cloned for in vitro and in vivo evaluation. So investigators at the Children's Medical Research Institute, under the lead of Dr. Leszek Lisowski, are testing those treatments in iPSC cells and are working on phenotyping mouse models. In January, we have planned our main efficacy study and long-term safety study, currently using 2 different routes of administration, intrathecal and intraventricular. And as mentioned, 2 different promoters. If these studies are going to be successful, we see a significant effect and no significant side effects, we will initiate a clinical trial in late 2023, early 2024. We have already initiated discussions with the European Medical Agency and national authorities, who are generally very supportive of this project and of this development. So when we started this 1.5 years ago, we had no idea we would go this far in such a short amount of time. At the beginning, I did not even know where to start. The articles I read at impossible hours were really hard to understand. I have reached over 300 researchers and would like to take this opportunity to thank them all for their sincere desire to help affected kids. And also, importantly, in Slovenia, research are working with us, building a center for technologies of gene and cell therapy. And so in the future, I have no doubt that genetic rare diseases will be one day a thing of a past. That is why I'm very proud and optimistic about the future of our children. Today we have a clear roadmap, amazing researchers and several ongoing gene therapy projects. And we are more determined than ever that we will not stop until we find something that will give our children a chance at life. And I believe that as with any new technology, the costs are astronomical now, but will come down to the process when the process will become more automated. Unfortunately, Urban does not have the luxury of time, so the money needs to be raised now. So this is also the way you can help Urban and 400 other kids diagnosed with CTNNB1 syndrome. You can donate directly through our CTNNB1 Foundation page or follow the link on the GoFundMe. We also have a PayPal account. You just type CTNNB1 Foundation. So I would like to really thank you for your attention, and please see my email for further questions. Thanks also to the organizers. I think it is important to have as many opportunities as possible to increase the awareness of the CTNNB1 syndrome and the current research activities. Thank you very much.